This is the last video in the cell division unit. In this video, we will discuss how the cell cycle is controlled and why that regulation is important to the health of the organism. Mitosis is necessary for growth of a multicellular organism, wound healing, or in the maintenance of actively dividing tissues, such as the skin or lining of the intestines. However, cell division cannot occur all the time because uncontrolled division is dangerous and can lead to cancer. This means that mitosis must be regulated so the cell only divides if new additional cells are required by the body. This regulation is controlled by a specific set of proteins called cyclins. The name of these proteins comes from the fact that they appear and disappear from the cell in a cyclical fashion. When a cyclin protein appears in the cell, it will bind to an enzyme called a cyclin-dependent kinase, forming the cyclin CDK complex. The cyclin-dependent kinase cannot function until it binds to the cyclin. Now that the complex is formed, it can interact with various cellular target proteins. The kinase enzyme phosphorylates the target by adding a phosphate group to it, causing an effect in the cell that controls mitosis. The cyclin protein is eventually degraded and the CDK goes back to being inactive. There are numerous different cyclins and CDKs that act at specific checkpoints along the cell cycle. Checkpoints are a point in the cycle where the cell can pause the cell cycle if something is not right, before progressing to the next stage. They are seen here as red circles to represent a stoplight, where the cellular processes stop before moving to the next stage of the cell cycle. Three major, well-studied checkpoints include the G1 checkpoint, which surveys the DNA to ensure that it is not damaged before replication. The cell does not want to replicate damaged DNA, because this would pass mutations on to the daughter cell. If all the DNA is undamaged, the cell will proceed through this checkpoint. The G2 checkpoint occurs at the end of G2 and checks to ensure that all the DNA replicated properly during S phase. The cell does not want to replicate if there is not a full set of replicated chromosomes to pass on to the daughter cells. If replication has occurred properly, the cell will progress through this checkpoint. The third checkpoint is the M checkpoint, which ensures all of the mitotic spindles have attached to chromosomes and they are all aligned at the center of the cell before progressing into anaphase. This ensures that the sister chromatids are all properly separated to each of the individual daughter cells. We can observe when the cell passes through this checkpoint, as that is when the sister chromatids are released from each other as anaphase starts. There are several different cyclin and CDK complexes that act at specific steps of the cell cycle, and it is these complexes that allow the cell to proceed through the checkpoints and complete the functions necessary for each stage of the cell cycle. For example, the G1S cyclin is produced towards the end of G1, which allows the G1S cyclin CDK complex to form. It is this complex that gets the cell ready for DNA replication in S phase and allows progression through the G1 checkpoint. An example of regulation at the G1 checkpoint is the protein P53. When DNA is damaged by radiation, a protein called P53 is activated by the addition of a phosphate group. The activated P53 leads to the inhibition of the G1 cyclin CDK complex, which prevents the cell from going through the checkpoint. This arrests the cell at the G1S transition, giving the cell time to repair the DNA damage before moving into S phase. P53 is an important protein because of its role in regulating the cell cycle. In fact, many cancers contain DNA mutations in the P53 gene, which prevents it from doing the proper regulation. Control and regulation of the cell cycle is so important because if the cell divides at times when the body does not require any new cells, this can lead to the development of cancerous tumors. Cancers occur because genes necessary for the cell cycle become mutated so that their encoded proteins no longer function properly. There are two types of cancer-causing genes: proto-oncogenes that are required for cell division to occur. They are like the gas pedal of the cell that says to go and divide. If they are mutated, it is like the gas pedal gets stuck down and the cell divides all the time. Tumor suppressor genes encode proteins that prevent the cell division from occurring. These are like the brakes of the cell. If they are mutated, the cell loses its brakes, so it can no longer stop cell division when it's no longer needed. Typically, mutations occur in both types of genes, leading to uncontrolled growth and cancerous cells. This multiple mutation model for cancer development is shown here. One mutation causes some abnormal changes in the cell, but the cell is still functioning properly. Another mutation then occurs in a different gene. For example, a mutation in the proto-oncogene causing formation of an oncogene, or a cancer-causing gene. Now the cell begins growing abnormally, but its abnormal activities are not harmful to the body overall. These are considered benign tumors because they are slow-growing and non-invasive to the surrounding tissue. As more mutations continue, a malignant tumor now develops, as the mutated cells are now growing rapidly and invading the rest of the tissue, preventing the organ from functioning properly. Eventually, the accumulation of mutations can lead to metastasis, where the tumor cells spread to other areas of the body. The gradual accumulation of mutations in multiple genes often occurs over a period of years.